thank you very much for that kind introduction, and I would very much like to thank the organizers of this meeting, both from FAPESP and also from the Office of the Vice Chancellor at UC Davis. I'm going to be talking about interdisciplinary research in neuroscience at UC Davis. I'll talk about some general ideas about the importance of an interdisciplinary approach to pursuing questions in neuroscience, and then I'll show you some new data from some research that we're doing as part of a newly funded interdisciplinary center at Davis. Neuroscience is one of the newest areas of the biological sciences, and it is at its core an interdisciplinary science. Neuroscience didn't exist as a separate area in biology until about 40 years ago, when people who were interested in studying neural function in areas such as physiology and anatomy got together and founded the Society for Neuroscience, which was initially a very small group. Over the years, people have traditionally identified with the particular areas of neuroscience that reflects the general methods that they're trained in and have developed. So typically people have identified as cellular neuroscientists or molecular neuroscientists or systems neuroscientists or cognitive neuroscientists or computational or clinical or translational. However, the reality is that this interdisciplinary science itself has become increasingly interdisciplinary, in large part because of the challenges that are raised in trying to address basic issues of brain health. And over the years, what we have seen is, as interdisciplinary teams have worked together, that there have been connections that have developed between labs. This may reflect shared trainees training in different methods, an increasing trend in neurosciences for people to do multiple postdocs, then also enable them to develop skills in different methods. And initiatives like the Brain Initiative, that I'll mention again in a moment, have brought together people working across different levels, so that now increasingly these boundaries between the traditional areas of neuroscience are breaking down, and what we're seeing instead is a big kind of small world network of neuroscientists working on grand challenges and crossing across the traditional methodological boundaries. In addition to building bridges across sub-areas of neuroscience, one of the great challenges now is bringing in people from outside of neuroscience. And increasingly what we're seeing is the integration of other areas of biology, such as genetics, other areas of science and technology, such as engineering and nanotechnology, physics, chemistry, mathematics. And this is the field right now where people are really experiencing the broadening of the boundaries of neuroscience, a rapid advance in technologies, and an increasing ability for interdisciplinary teams to tackle fundamental questions about how the brain works and how it can sometimes go awry in brain disorders. Harris Lewin mentioned briefly the Brain Initiative. This is a very exciting development in the United States, an initiative that came out of the White House, likened to the challenge of putting a man on the moon, figuring out how to actually understand how the human brain works and how we can use that knowledge to get fundamental answers as to the causes of disorders such as autism, schizophrenia, Alzheimer's disease. The goal of the Brain Initiative is largely a technical one. It's to accelerate the development and application of new neurotechnologies that will allow us to understand the dynamic function of the human brain and how it integrates neuronal and circuit activity over time and space to lead to our experience, our thinking, our emotions, and also to develop the tools that we need in order to be able to diagnose and ultimately treat the serious brain disorders that affect so many people in our society. And one of the more exciting aspects of the Brain Initiative is it's brought in some new dollars from the federal funding agencies, the NIH and the NIMH, but it's also brought in dollars from agencies that haven't traditionally invested in neuroscience. This includes the Department of Defense, as well as a number of important foundations. Harris mentioned that at UC Davis we have a number of interdisciplinary neuroscience centers. I direct the Center for Neuroscience. This center has currently 23. We're in the process of recruiting our 24th faculty member. Our faculty work across all areas from cellular, molecular, systems, cognitive, computational, and translational neuroscience. They frequently organize themselves into interdisciplinary teams, and they have a number of areas of strengths, including developmental neurobiology, synaptic biology, neuroplasticity, learning and memory, sensory systems, and translational neuroscience, particularly a focus on the neurobiology of autism, schizophrenia and bipolar disorder, and Alzheimer's disease. In addition to the Center for Neuroscience, though, Harris also mentioned that we have actually 3 interdisciplinary centers that focus on different aspects of neuroscience. So our center, which we typically refer to as the CNS, is a basic neuroscience center largely, and focuses on all areas from cellular through systems, computational, and translational. The Center for Mind and Brain is more of a cognitive neuroscience center. It's more psychologically oriented. The Mind Institute focuses on neurodevelopmental disorders, particularly autism. And we have this interdisciplinary graduate group that kind of binds together these centers, and it very much helps to facilitate crosstalk between them and to create that kind of network that I showed you in my earlier slide. All in all, we have probably 150 neuroscientists broadly defined working at UC Davis. In addition to these 3 centers, they're found in a number of departments and colleges across the whole campus. And again, our neuroscience graduate group, that interdisciplinary group, is one of the ways that that community gets bound together. To foster interdisciplinary research, one needs opportunities and incentives. I think that's why we are all here today. And Harris mentioned the RISE initiative at UC Davis. I will mention that one of the interdisciplinary groups that was funded very generously by this initiative was a neuroscience-oriented group. And that additional funding, which actually amounted to about $800,000 over a 3-year period, enabled us to do some very risky research, high risk in terms of risk for failure, very quickly, and allowed us to build a portfolio of preliminary data that allowed us to compete for this very important NIMH Conti Center that we recently received funding for. And what I plan to do during the rest of my talk is to talk a little bit about that cool data that we collected that was funded by the pilot funds from the RISE, and that helped to convince the skeptical reviewers at the National Institute of Mental Health that they should give UC Davis one of these Conti centers. So the PI of the RISE initiative was Kimberly McAllister, a developmental neurobiologist in the Center for Neuroscience at UC Davis. I'll show her picture a little later in my talk. Our center has been organized around the notion that neuroimmune mechanisms might play an important role in psychiatric disorders. Okay, so here's the team, and this is Kim, who was PI of the RISE. What's very exciting about this group is that we have members from the whole diversity of neuroscience on campus. David Amaral, to my left. I'm not sure if this pointer is working. Can you see it? Okay, well, so we'll just go from left to right, starting at the top. Kim McAllister, Simon Cherry from Biomedical Engineering, myself from the bottom, David Amaral from the Mind Institute, Melissa Bauman from the Mind Institute, and then we brought in Dan Geschwood from UCLA to help us with our genetics. This center is focused broadly on questions of the role of neuroimmune mechanisms in psychiatric disorders, but specifically on the problem of schizophrenia. Schizophrenia, for those of you in other areas, is a very common and very serious mental disorder. It affects roughly 1% of the population. In a big city like São Paulo, with, I understand, in the greater area, maybe 20 million people, you have tens of thousands of people with this illness. Early intervention can make a big difference with this illness, but many patients continue to have poor outcomes with high levels of homelessness, unemployment, incarceration, substance use. It has its onset in late adolescence, and it's caused by some complex of gene-environmental interactions which we are still struggling to understand. And this particular center is focused on one potential important environmental risk factor. Unfortunately, although we do have therapies for this illness, treatment at this time is symptomatic, and it has limited effectiveness for many people with the illness. So there's a strong and driving need to develop new therapies that don't just treat the symptoms, but actually tackle the underlying causes of the illness. Now we know from imaging and other studies, using tools that have become available to us over the last 20 years or so, that there are subtle changes in brain anatomy that are present in this illness, that using EEG and MEG and other measures we can obtain evidence of altered neurophysiology. Circuits that are involved in the control of attention and memory are disrupted in the illness, and that seems to be associated with an inability to mount oscillatory activity in local circuits in the illness. And post-mortem studies suggest that there are changes in connectivity at the neuronal level with reduced numbers of dendritic spines in key areas of the brain, such as the dorsal prefrontal cortex. What we don't really know, and where we can use methods such as PET scanning to show that there is increased subcortical dopamine that's present and associated with psychosis in the illness. So we know a lot at the systems and circuit level about what's going on in this illness, but what we don't understand is at the cellular and molecular level, what is the cause? So this takes us to this rather surprising hypothesis. Probably 5 years ago, a lot of people would have thought that this was kind of a crazy hypothesis, that neuroimmune mechanisms could play a role in the underlying pathophysiology. But in fact, we do have growing evidence that schizophrenia is a developmental disorder. People who develop the illness show early signs of atypical development that are subtle, but nonetheless do predict later development of the illness. We also know that MHC and cytokines play a very critical role in normal brain development. In addition, they play a role in adult synaptic function and in the development of the balance between excitation and inhibition in the cortex. This is a relatively new finding from basic neuroscience. About 12 years ago, Carla Schatz at Stanford discovered that in fact some of these molecules were present in the brain. People thought that the brain was immunologically privileged and was devoid of these molecules. And it's since been shown, and Dr. McAllister in our center has been one of the key people to demonstrate this, that these molecules actually play a very critical role, both in guiding early brain development and in stabilizing and managing synaptic connectivity of the kind that has been shown in these post-mortem studies to be reduced in schizophrenia. One of the other observations that has sort of captured people's attention about the potential role of neuroimmune mechanisms is the fact that maternal infections, such as influenza during the late part of the first trimester, is associated with a 4 to 7 times increase in the incidence of this illness in the offspring. When one studies the immune system in schizophrenia and measures molecules like cytokines in the blood or the CSF, there are consistent alterations. And in a subset of people with schizophrenia, one can measure these changes also in postmortem brain tissue analysis in people with the illness. And more recently, PET ligands have been developed that can index markers of neural inflammation in the form of activated microglia in the brain. And there are now a couple of studies that show that there's an increased signal in young people who've recently developed schizophrenia using this method. And then perhaps one of the more surprising findings of all is recent genome-wide association studies that have begun to identify initially a cluster of several and now actually up to about 108 genes that are associated with schizophrenia at the genome-wide level, have shown that by far the strongest signal is from a molecule, from a gene that codes an area on a chromosome that codes MHC, the major histocompatibility complex. A very surprising finding that on its own is particularly curious, but when put together with these other observations, begins to build a case for trying to understand whether neuroimmune mechanisms actually do contribute to altered brain development and lead to increased risk of schizophrenia in humans. More recently, people have developed animal model systems, and I'm going to quickly describe one of these. These are models of maternal immune activation that lead to offspring. I'll talk about a mouse model and also a non-human primate model, and I'll do that in 5 minutes. I'll do my best. And these animal models have altered neurodevelopment such that after a period of fairly typical development, they then begin to show changes in social behavior, changes in cognition, and other phenotypic alterations suggestive of those that are seen in people with schizophrenia. And then finally, there are a number of small clinical trials now using anti-inflammatory medications that do show some impact on the symptoms of the illness. So this is kind of the background for this big project. Our goal is really to test the hypothesis that altered signaling in these molecular pathways affects brain development, the development of normal synaptic connectivity, the maintenance of normal synaptic function in adulthood, and leads to the emergence in changes in brain connectivity and behavior that underlie disorders such as schizophrenia. And this is sort of a schematic of what I just said. I'm going to skip ahead because I just want to very quickly describe our overall approach. Essentially, we have parallel studies in humans, people with recent onset psychosis, a mouse model of maternal immune activation, and then a very novel non-human primate model. And our key questions that I will now focus on in the next couple of minutes that really were the ones we tried to address through the RISE project that provided the preliminary data for our Sender grant were, is there evidence of brain inflammation in first episode patients with schizophrenia? Can we measure this using novel methods from MRI? And then, does the non-human primate model of maternal immune activation, which does show this behavioral phenotype with a normal period of development followed by subtle changes in social behavior, followed by more severe behavioral disturbances as the monkeys go into adolescence. Is there evidence using molecular imaging methods that could link this particular animal model to the clinical phenotype of schizophrenia? I'm going to skip through the methods here. I'll just tell you we used a new method of diffusion magnetic resonance imaging that allows us to get a coarse measure of brain inflammation, and that is free brain water. And we used this technique in people with schizophrenia. A previous study had shown increases in this signal in a chronic group of patients. And in our first episode patients, we did indeed see increased whole brain water, suggesting that these young people who are going through their first episode of psychosis do have an inflammatory process that's happening in their brain. And then in the monkeys, we did 2 PET studies in quick succession. Just as these monkeys were going into adolescence, when they were showing evidence of developing these behavioral phenotypes that we would link to an animal model of psychosis or psychosis risk. The first method looked at a marker of inflammation, the same one that had been used in those clinical studies that I mentioned a few minutes ago. And what we saw was that in the grey matter, indeed, there was evidence for active inflammation in the cortex in those monkeys. And then we asked the question, do these monkeys have increased subcortical dopamine levels? This is a kind of very well-established molecular phenotype for psychosis. It's seen in people at different stages of psychosis, and it correlates with both the level of the psychotic symptoms and also the tendency for patients to respond to dopamine-blocking drugs. We used a ligand that has been used widely in patients to measure this signal in the basal ganglia, and what we saw was that in fact these monkeys do have evidence of increased subcortical dopamine, this molecular phenotype, to go along with their behavioral phenotype, and to provide some validation of this non-human primate model as one that may be related to enhanced risk for psychosis. All right, so just quickly to summarize. Integrative neuroscience will accelerate progress towards more effective treatment and cures in psychiatric disorders. And this does require interdisciplinary teams using cross-cutting approaches to pursue shared goals. And I think our success at UC Davis is testimony to our institution's commitment to making those sorts of investments. With regard to the specifics of our project, there does seem to be increasing evidence that neuroimmune mechanisms may mediate the effect of environmental risk factors. So we looked at one, which is maternal infections or maternal immune activation, but other environmental risk factors associated with schizophrenia, including head trauma, psychosocial stress, and drugs, also lead to changes in brain cytokines and might result in similar altered changes in brain development that in turn might lead to schizophrenia risk. I think there's growing evidence that altered neuroimmune signaling, such as cytokines and MHC, might lead to this atypical brain development. And our goal in this newly funded center is to understand these mechanisms so that we are able to develop a new set of imaging biomarkers that can be used to actually identify the illness during its earliest phases, and also new treatments that may target pathways that are altering brain development in a way that's much more proximal to the causes of the disease, rather than the current treatments we have, which really deal with the end result of altered brain development and increased subcortical dopamine. So I will just end by saying that I must acknowledge the participants of our research, particularly the young people with early psychosis who participate in our early psychosis prevention and early intervention clinic in Sacramento at the UC Davis Medical Center. And I understand from talking to Dr. Brustad actually that you're going to hear more about that in the next talk, and that he and I actually have quite a lot in common in this regard. And thank you very much for your attention.